how do they interact, so then we can do a bridging, and then we'll know how molecules move in. Yeah? And these molecules
express the gamma delta T cell receptors, but out of which the dominant subset is the V gamma 9, V delta 2, and you have a small subset which is V delta 1.
Are important for signaling in plasma cells. It's absolutely unclear. But just for the sake of completion, I show you that if you act through SH2, you can act PI3 kinase, AKT.
The thymic hydrocellularity defect that's seen in harmful mice. DN3 and DN4 cell proliferation as measured by EDU and PRDU incorporation.
Of what immune regulatory mechanisms are. These are mechanisms that are important to maintain immune homeostasis, and also mechanisms that are important and operate to counterbalance perturbed immune response, and this is the case.
During the class, that I'll be talking precisely on some of the HIV-1 is a highly evolved virus. Just few lines. HIV is a huge public health problem.
Actually emphasize something. Is that we have been talking about a lot of things that predispose us to autoimmunity, but we never talk about salt. And if you really look at food that we eat, so average requirement for food is about 35 milligrams of sodium chloride per day. So I don't want to alarm you guys, but I see a lot of KFC and a lot of McDonald's popping all over India. I do not know whether it's a good thing or a bad thing. But I want to emphasize one thing, is that of all the T cells, it looks like HTK1, a kinase that regulates the salt channel, is crucial for differentiation of Th17 cells. A modest increase in the salt in diet or exogenous addition of salt to the media immediately induces naive T cells.
I am presenting today instead of Professor Noah Kaisakov, who could not manage to come for some reason. So any hard question will be directed to Professor Noah Kaisakov. Today I am going to talk about the regulation of crack eruptor protein in T cell signaling. I'd like to start acknowledging my mentor, Professor Kaisakov, and my wonderful lab colleagues who helped me a lot during my stay in there. CRAC stands for chicken tumor virus stem regulator of kinase. It was initially identified as a retroviral oncogene in late 80s. And the mammalian homologs or mammalian counterparts of CRAC are CRAC1, CRAC2, and CRAC-L. Structurally, CRAC1, CRAC-L are almost identical. They share almost 60% identity in amino acid, even ubiquitous in nature, has been tested in different organs and tissues of mouse. However, in some cases, in some parts, the expression are more specific to some organs. Crack eruptor proteins are found to be functional downstream of the TCR drug 70 complex. Depending on the antigen being presented to TCR, crack binds to different binding partners like SOS, SOAN, C3 gene, LOC1HP, and activates the MAP kinase signaling pathway, activating the transcription factors like NF-kappa B and AP1 together with the force sensor. While dissecting the structure of CRAC2 molecule, we found the presence of both SH3 binding site, the polyproline rich region, and SH2 binding site at the tyrosine 2 conjugal position. And also, we found a regulatory proline at 237 position in the context of lysine, proline, and isoleucine, which make the molecule an potential substrate for PPIs. PPAs are the peptidyl proline C-star isomerase enzyme that changes the conformation of protein post-translational. hypothesized that CREC2 could be a substrate, in vivo substrate of PPIs. And if so, there should be two distinct populations of CREC2 in vivo. One is in trans conformation, that is an open conformation, where CREC can now associate with different binding partners, both through its SS2 and SS3 domain, and transmit the signals. Now, another conformation is cis conformation, where CREC is in closed conformation, and it cannot now bind to different binding partners, at least some of them, and hence the sum of the signaling events, the signal transactions events are disabled. So to test this hypothesis, C3G is one of the binding proteins of CREC, which binds to CREC in constitutive manner. In densitometric analysis, what we found in white bar, the amount of CREC1 being precipitated with C3G remained almost identical, both in treated or non-treated cells. Whereas in the case of CREC2 in black bar, the amount of CREC2 pulled down by C3G decreased almost to half when the cells were treated with cyclosporine A and FIMSA. In this experiment, we prepared subclones of darker cells which are overexpressing MIG-TEC CREC1 and MIG-TEC CREC2. And we did the treatment of cells with CSA and FK506 and activation with OKT3 for 1 minute. When we did an anti-MIG immunoprecipitation assay and tested for the presence of C3G, we found the amount of C3G being immunoprecipitated with CREC1 remained almost identical. But where the amount of C3G precipitated with CREC2 leveled up when it was treated with CSA and FK506. A transfer migration assay was then conducted. It's one of the functional assays that is dependent on CREC and C3G association. And we did it with the darker cells and the two subclones which are overexpressing MIG-TEC CREC1 and MIG-TEC CREC2. So what we found was a significant increase in green bar of cell migration when that was overexpressing CREC1 and CREC2. But in case of darker CREC2, when the cells were treated with drugs in red bar, the migration decreased significantly in case of CREC1. There was a decrease in migration in case of here, one of the PPIs inhibitors, cyclosporine and FK506, both are called as immunosuppressive drugs also, inhibit the association of CREC2 with C3G in darker cells. However, the drugs cannot inhibit the association of CREC1 with C3G. And the migration is enhanced in darker cells, overexpressing both CREC1 and CREC2. But it's reduced upon the treatment of cells with the immunosuppressive drugs. A very short announcement about the postdoctoral position in our lab. Whoever is interested, please feel free to contact. Thank you very much for your attention.